So I have pretty ambitious plans for today. Let's tackle it. We're going to talk about various biogeochemical cycles. We're going to talk about the requirements for growth. We're going to briefly describe Campylobacter jejuni infection. We're going to do a lab. And then I hope to tackle the methods of microbial control. So let's start without any further ado with biogeochemical cycles and nutrients first and foremost. So elements found in the living tissue, obviously carbon, hydrogen, and oxygen. The sources, possible sources, CO2, water, organic compounds. Other important elements that can be found in various biological macromolecules. Nitrogen, which is found in the nucleic acids like DNA and RNA, and of course in the proteins. Sulfur, found in proteins, and phosphorus, that is found in DNA and RNA and phospholipids. Now, what I want you to appreciate here is that we're going to talk specifically about carbon, nitrogen, and sulfur. All those elements, they kind of cycle between the organic compounds and inorganic ones. And you will see the common theme between all of those different biogeochemical cycles. So we're going to start with the carbon cycle. And the first reaction is going to look a hell of a familiar reaction. Carbon dioxide and water, in the process of photosynthesis, will form carbohydrates and oxygen. Okay. Good. Now, what I want to highlight is that this specific part here is inorganic. This specific part here is organic. So in the process of photosynthesis, inorganic carbon is turned into its organic form. And in the process of cellular respiration, so that's going to be photosynthesis. And in the process of cellular respiration, organic carbon turned back into inorganic form. Now, does that make sense? That's what we were talking about before, right? So that is sort of a one big staples of carbon cycle. But there are a couple of other types of microorganisms that participate in carbon cycling as well. One of them is using carbon dioxide as a final electron acceptor, essentially converting it into methane. So these microorganisms form methanogens. Okay, right here. I'm going to highlight some of this stuff. Now, here's the problem. Here's the problem with methanogens. They generate a huge amount of methane, which is a significant contributor to greenhouse effect. You know, the greenhouse gases, CO2 is the most famous greenhouse gas. In fact, methane is much sort of more, has much stronger greenhouse effect compared to CO2. So methanogens are commonly found in the soil, in the swamps, in the ruminants of, in the guts of ruminants such as cows. And yes, indeed, cows are one of the main contributors to the global warming, okay, to the production of CH4, methane. Now, can we do something about that methane? Can we somehow get rid of methane? Turns out, yes, there are microorganisms that use methane as the source of carbon, and they convert it into organic compounds, other organic compounds. For instance, pyruvate, and these microorganisms are called methanogens. So while methanogens, they kind of contribute to global warming, albeit inadvertently, methanogens allow us to sequester methane and reduce the amount of greenhouse gases. Now, I want to highlight this. Look. This is an oxidized form of carbon dioxide, of carbon. This is a reduced form. This is oxidized form. This is a reduced form. This is a reduced form. This is partially oxidized. I'm going to put a little note here that this is reduced. And this color will mark oxidized. So you can see that, in fact, the cycling occurs between the oxidized and reduced form, reduced and oxidized form. Does that make sense? So that whole thing, the whole idea with the cycling is about reduction and oxidation, reduction and oxidation. Good? Even more sort of pronounced, this effect will be when we will talk about nitrogen cycles. Nitrogen, as you can see from here, is essential for a bunch of biological macromolecules. But nitrogen itself, that is in the atmosphere, is extremely inert gas. It does not get into the chemical reactions very easily. So converting nitrogen into sort of chemically active form has always been a pain in the ass. Humans use so-called heavy wash process, which requires extremely high temperature, high pressure, very expensive catalysts. Not a great process, to be honest. Microorganisms managed to do the same process much more economical and efficient way, which we cannot replicate so far. So the first step is for microorganisms is to convert nitrogen into ammonia and then incorporate ammonia into organic compounds. Now this process right here is called ammonification. Okay. So essentially in this process, nitrogen is reduced. We've got a reduced form. Now when these microorganisms or macroorganisms, doesn't matter, and they die, this nitrogen then is oxidized. Okay? In the process of, well, various takes of cellular respiration during decomposition. It is oxidized to nitrite, and then from nitrite to nitrate. Okay? So this whole process, both of these arrows, I'm going to do this, is called nitrification. Now, what humans do, this nitrates, nitrites, they can exist in the form of various salts, for instance, ammonium nitrite, nitrate, or potassium nitrate. And these compounds can be used as a fertilizer. Turns out, in many microorganisms, nitrate may serve as the final electron acceptor. Now look at this. You can essentially use nitrate and nitrite as the nitrogen sources as well. This is why we use different nitrites and nitrates as the fertilizers. Does that make sense? But here's the problem. You have fertilizers, and these fertilizers end up in your water supply, okay? Lakes or rivers. They can cause an abundant growth of various microorganisms, which will essentially deprive river or lake from nutrients. Does that make sense so far? And we call it blooms. Algal blooms can be other microorganisms, all right? So in order to remove nitrates and nitrites from agricultural runoff from the fields, we actually, in the water treatment plants, we use microorganisms that are capable of the process known as denitrification. Okay, so if we will go through this process one more time, in aminification process, nitrogen is reduced to ammonia and amines. During decomposition, which is essentially respiration, amines are oxidized to nitrites and nitrates. And then nitrates, in the process of denitrification, are reduced into nitrogen. And here you have a perfect cycle. Does that make sense so far? Good? Okay. Here. Because I'm trying to occupy as much space as I can. So I'm going to move over here, this side of it. And I'm going to talk about finally sulfur cycle. The sulfur cycle is really a relatively simple idea. So as you may remember, we mentioned that sulfate may serve as the final electron acceptor in the process of anaerobic respiration. The sulfate will be reduced to molecular sulfur, like the solid sulfur, that can be deposited, and eventually to hydrogen sulfide. Okay. So you all are familiar with that when you feel the smell of rotten eggs. They don't just rot. Usually they rot because the microorganism that you're definitely familiar with, Salmonella typhi or Salmonella enterica, will replicate in the eggs, and those microorganisms actively produce hydrogen sulfide, and voila, you're going to have hydrogen sulfide. Okay, and I'm going to put not just hydrogen sulfide, I'm going to put SH, so organic, sulfur, But then, of course, in the process of decomposition, this SH, the thiol group, can be oxidized to sulfate, and hydrogen sulfide can be used as an electron donor in various, processes of like anaerobic photosynthesis, for instance. Okay, I'm going to give some. So let's do anaerobic respiration. Oxygen photosynthesis. And this is decomposition. Now, look, if we will highlight, this is oxidized form, and this are reduced forms. Does that make sense so far? Yeah, definitely with a better camera. Okay, we're good? Yana? Keeping up? Good. All right. So before we will feel the contents of that amazing stuff, I want to chat about microbial communities, okay? And let's give it a name: communal relationships. Now, what do I mean by communal relationships? So microorganisms, they very rarely live by themselves. I hope that makes sense, right? So the relationships between microorganisms can be obligatory and can be non-obligatory. Obligatory means that microorganisms, to a very large extent, depend on those relationships. Does that make sense? So one microorganism, for one or another reason, cannot exist without another. Okay? So we're going to put here obligatory, and this will be non-obligatory on the right. Okay, so there are three obligatory relationships that I want you to know. First is mutualism. So in the mutual relationships, in mutualism, you have two microorganisms, A and B, and they depend on each other. For instance, microorganism A produces a nutrient factor that microorganism B requires. And microorganism B, in turn, conditions the environment so microorganism A can survive. Does that make sense? If you get rid of one of them, the whole system goes belly up. Okay? Second one is commensalism. In the commensal relationships, a commensal, which is going to be B, benefits. And cohabitants A isn't harmed or doesn't benefit. Does that make sense? To kind of assuage any questions, in the lecture notes, I provide an example of commensalism, which is satellitism. It's a nutritional commensalism, meaning that in satellitism, for instance, it's in the notes, meaning that microorganism A produces an essential nutrient for microorganism B. Does that make sense? That's a specific example. So satellitism is a special case of commensalism. But commensalism can be much more than just nutrient-based relationships. Does that make sense? Good? Parasitism. Two microorganisms and parasite A here cannot survive without B, but at the same time, parasite A hurts B. Does that make sense? Now, if we're talking about microorganisms, the concept of hurting a little, weird, but say microorganism A uses metabolism of B for its own advantage, so that replication, for instance, or survival of microorganism B is impaired. Does that make sense? On the broad spectrum, all human infections are examples of parasitism. Make sense so far? I don't know why I'm looking at the camera. Notice that. We're good? Okay. Two non-obligatory relationships that I want you to know. Synergism and antagonism. Now, I really don't know what picture to give you for synergism. Okay, so A and B, they both can grow independently. Does that they don't need each other. But when they are together, the survival of each significantly increases. Does that make sense? So I deliberately don't put interdependence. And in antagonism, you have two organisms. They can grow without each other, but when they together, they are mutually exclusive. They kill each other. It's not even parasitism. They just destroy each other. Does that make sense? We're good. Any questions? Yes. No. Oh, for biogeochemical cycles, you need to know sort of terminology. Let me explain. For instance, if I ask you what happens in a cellular respiration, inorganic carbon is converted to organic, fixed into organic. What happens in the photosynthesis? The other way around. What do methanogens do? Produce methane from inorganic CO2. What methanotrophs do? Convert methane into other organic compounds. What is denitrification? Conversion of nitrate into nitrogen. What is ammonification? Conversion of nitrogen into ammonia and organic nitrogen. What is nitrification? Things like that. What happens during in regards to sulfur cycle? What happens in oxygenic photosynthesis? Hydrogen sulfide is converted to sulfur. What happens during anaerobic respiration in regards to sulfur? Sulfate is converted to hydrogen sulfide. Did I explain? Am I clear? Good. Kind of wink-wink thing, but I think there are like 2 or 3 questions only on biogeochemical cycles, and they're not complicated at all. It is very, very straightforward. Okay. So while I'm getting rid of this stuff, your exam is going to be 2 weeks from now on the unit 3. If we do everything right, we should be able to cover review antibiotics next week, and that'll be it. It's really not very hard. Uh-huh. You don't use for cows specifically, but you can, so basically, especially in the places like on a farm, methane is actually produced when, what's the word, manure decomposes, whatever. So you can basically add methanogens to neutralize the methane that is being released during the decomposition. And methane is a huge problem for landfills. So we don't do it, but it would be lovely to collect the methane and convert it into some organics without releasing it into the environment. That makes sense? Okay, so we're going to talk about the growth requirements. And I'm going to start with oxygen requirements for growth. Now, in the lecture notes, there are lovely pictures of microorganisms growing at the different locations in the tube. I'm not going to draw it. I'm going to address slightly different things. I'm going to address the whether or not microorganisms in a particular group require oxygen and what kind of metabolism they use predominantly. Okay, so we're going to start with an obvious choice. So first one is obligate aerobic microorganisms. So obligate aerobes, they need oxygen. And metabolic pathway that they use, quite obviously, aerobic respiration. Second group is obligate anaerobic. Now, for these microorganisms, oxygen is toxic. So these ones usually use fermentation or anaerobic respiration. Third group is air-tolerant anaerobic. Now, to differentiate them from obligate anaerobes, I'm going to write this. They don't need oxygen. Can they survive in the presence of oxygen? Yes. Do they use it? No. Air-tolerant anaerobes, by the most part, are fermenters. But some also can use anaerobic respiration. Probably the trickiest group here is facultative anaerobic. The word facultative means they can, but they don't have to. Does that make sense? They can, but they don't have to. So they can use oxygen. Well, in fact, they use oxygen when it's available, okay? And they switch to fermentation when it's not. Because remember, in aerobic respiration, you have glycolysis, and then Krebs cycle and electron transport chain. If oxygen is available, they will run the whole gamut. But if oxygen is not, they will switch to fermentation immediately after glycolysis. So we can write down this. They use aerobic respiration with oxygen and fermentation without oxygen. Okay, we good? And the last one is microaerophile. Microaerophiles are microorganisms that require a very specific oxygen concentration. Usually much lower than atmospheric oxygen. Okay, we call them Goldilocks. Okay, that makes sense. Goldilocks. So if you will put them in a completely anaerobic environment, they will not grow. If you will put them in the normal atmosphere, they won't grow. They need that just right amount. And they respire aerobically. Okay, we're good? Good. Now, there's a interesting sort of side effect of respiring and being exposed to oxygen. Presence of oxygen leads to the formation of reactive oxygen species. Of this, there are two of them, most important. It's superoxide anion and hydrogen peroxide. So both of them are toxic for a cell. Okay, superoxide anion is extremely toxic. Hydrogen peroxide is a little bit less toxic. So cells that would like to either be able to grow in the presence of oxygen or use oxygen for respiration have to be able to deal and to neutralize those reactive oxygen species. Does that make sense? You're all well familiar with hydrogen peroxide being used as antiseptic against bacteria. Makes sense, right? Here's the thing. The enzyme that neutralizes superoxide anion is superoxide dismutase. The enzyme that is used to neutralize hydrogen peroxide, two enzymes, catalase and peroxidase. I'm going to give them numbers, 1, 2, and 3. So look at this. If you have obligate aerol, microorganism that not only constantly exposed to oxygen, but also actively respires. This microorganism has to have all three enzymes to neutralize reactive oxygen species efficiently. Does that make sense? 1, 2, 3. If you have obligate anaerobe, it kind of trades it. Since it is unable to survive in the presence of oxygen, it has none of those enzymes. None of them. Okay? Now, air-tolerant anaerobe, it does not respire, so there's no requirement for superoxide dismutase. Okay? But hydrogen peroxide will be inevitably generated. So it's got to have at least one enzyme to neutralize hydrogen peroxide. Most likely, it's going to be number 3. Now, since facultative anaerobe can respire, 1, 2, 3. Microaerophiles. It can depend. I'm going to put this whole thing in parentheses. So the exact combination of enzymes that this microorganism can have can vary. Does that make sense? By the way, did you notice that I've only used 7 months? Oh, much better. Okay, we're good? Last term in regards to kind of gas requirements, just the term. Capital file. Microorganism that requires CO2. Okay. All right, now next topic of discussion, sort of next requirement. It will be temperature. Something that I want to make very, very clear. I will not ask you to memorize the exact temperature ranges for each class. You have to have a general appreciation, sort of main features, okay? So we're going to start with a group called Cyclo Files. Cyclo Files basically can efficiently grow in the fridge. Below 40 degrees Celsius. Does that make sense? That's Cyclo Files. Now, second group will be Cyclo Trolls. Now, they grow at the room temperature. Slowly grow in a fridge. Why this is important, why we kind of put this microorganisms in a separate group. When you put your food into the refrigerator, you expect microbes to stop growing. Does that make sense? You can't expect it with cyclotrophs. They will continue growing. And they're responsible for food spoilage. Microorganisms like Listeria or various bacilli. Does that make sense? So you put your salmon in the fridge and you take it out 2 days later and you think, oh, it's fine. But Listeria kept growing and now it reached the amounts that it can be, a disinfection. Does that make sense? The most boring group. Mesophiles. 37 Celsius. Temperature of the human body. Okay, does that make sense? 99% of human pathogens belong to mesophiles. Are we clear? Now, 2 groups here. Thermophiles. I mean, 45 to, 80 degrees Celsius. So they kind of put it this way. They grow at the temperature of scalding water, not boiling, scalding. Are we clear? And finally, sometimes extreme thermophiles or hyper thermophiles. Lower the temperature of boiling water. Now imagine this. I ask you, Thermus aquaticus, its optimal growth temperature is 78 degrees Celsius. Which group does it belong to? I will never ask you to distinguish between thermophiles and hyperthermophiles or extreme thermophiles. Does that make sense? We kind of put them all together. Okay, we're good? Questions? Okay. pH requirements. This is, quite frankly, I consider it 3-point in the exam or taking candy from a baby concept. You have acidophiles, neutrophiles, and alkylophiles. Okay? So neutrophiles, pH 7, ballpark plus minus, so those are human pathogens. Does that make sense? Acidophiles, usually pH lower than 5. There are some extreme acidophiles, like the ones that grow in the sludge from the copper mines. And alkylophiles, up to pH 11, they can grow. Now, here's the deal. Look at it. Why it's kind of challenging to grow in the acidic environment. Acid can denature proteins. Does that make sense? Why it is challenging to grow in the alkaline environment? Remember cellular respiration process? More or less, electron transport chain. It depends on hydrogen gradient. Remember that? You put them in the base, there is no hydrogen gradient anymore. It's gone. We clear? Make sense? Moving on? Okay. Osmolality. So when we say osmolality, we refer to the environments with a high concentration of solutes. Does that make sense? Now, this is basically a terminological issue. Osmophiles is a general name. Doesn't matter what type of solutes we are talking about. For example, if you leave jam on the table, or jelly, for a couple of days, it's going to go sour in a couple of days. Jelly, like bought in the store. My wife tends to leave it on the dinner table all the time. Huh? Even if you leave it open, it's going to probably get like really bad on the surface, like tried out, but it's not going to go, like you're not going to have microbes growing. Does that make sense? Why? High concentration of sugar. It's very hyperosmotic environment. Does that make sense? Now, in that group osmophile, you have a subgroup that is called halophiles. Specifically, high concentration of salt. Does that make sense? So I want to make it very clear. All halophiles are osmophiles. But not all osmophiles are halophiles, because this is a subgroup. We're good? Okay. A couple more terms. I'm going to line them up. Just terms. Xerophiles, dry environment. Chlorophylls, high pressure environment. And finally, there are many terms: extremophiles, hyper-extremophiles. Let's call them hyper-extremophiles. Radiation, pH, temperature, etc. What do I mean? All of that shit together. Okay? I give an example of microorganism called Deinococcus radiodurans in the lecture. It lives in the cooling circuits, you know, the water that circulates in the nuclear reactor. And the most recent experiment that concluded, I just cannot not to share this. Give an idea. The plates with Deinococcus radiodurans were placed outside on the walls of International Space Station in the freaking space for 3 years. Vacuum, negative 270 degrees Celsius. When they took them in back, microbes kept growing, which is insane. They are like super resilient. Okay? Does that make sense? Now, the last bit before we go on break, because this is what I suggest. We're going to do this last bit, go on the break, come back into the lab. Okay? So this last bit will be about a microorganism called Campylobacter jejuni. Okay. Gram-negative, microaerophile, capnophile. Okay. Does that make sense? Leading cause of bacterial gastroenteritis. Do I need to describe the symptoms of gastroenteritis? Yeah. Fever, vomiting, diarrhea. Can last up to 2 weeks in some cases. The complication, sometimes Guillain-Barré syndrome. It's the which is autoimmune condition which leads to demyelination of motor neurons and paralysis. Usually, Guillain-Barré syndrome is transient. Can I have a question? Usually, Guillain-Barré syndrome is transient. In some folks, it can be like permanent damage. Okay. We're good so far? Transmission, it's a zoonotic disease. So, milk, poultry, eggs. Okay, this is why we pasteurize milk. This is why we cook eggs. This is why we cook poultry. One of the reasons. Okay. And last bit that I wanted to mention. So, oral Campylobacter species may be involved in inflammatory bowel disease. Now, there was some confusion about this. Let me explain. Campylobacter jejuni is a pathogen. Am I clear? if you drink milk contaminated with Campylobacter jejuni, you have a good chance to develop bacterial gastroenteritis. Am I clear? You do have some Campylobacter species in your mouth. Overgrowth of those Campylobacter species in the mouth is associated with inflammatory bowel disease. It is not C. jejuni. Does that make sense? C. jejuni does not cause IBD. Causes a completely different story. Oral Campylobacter. Do they? We don't know. There is an association of oral strains with IBD. Am I clear? We're good. Okay, cool. Awesome. I'm going to leave it at that. I'm going to pause the recording now. I'm gonna pack this chapter with a few concepts. I'm gonna put a little note to myself here to talk about it at the very end, because I want to get some stuff out of our way, and that's the methods of microbial control, basically cleaning agents. Okay. So first, before you have to clean up, you would rather contain. There are 4 levels of containment in the United States. I'm going to go from the strictest to the, from the easiest to the strictest. BSL 1, we are dealing with indigenous pathogen, meaning it's not exotic, it's not coming from some other continent or something. Very low virulence. So basically, we work at BSL 1 level. Okay, microorganisms cause virtually no disease. Does that make sense? Then we have BSL 2. It's probably the most common level of containment. Microorganisms are indigenous. Okay, mild disease, vaccine, and or treatment of it. Okay, then we have BSL 3. At BSL 2 level, you work in the biosafety food, not like this, but kind of similar. So you gotta have some protective gear. Usually it's gloves and a gown, no mask. BSL 3 may be exotic, meaning that it may come from some other location. So BSL 2 example would be rhinovirus. It's common cold. BSL 3 may be exotic. Illness can be severe. Usually vaccine and or treatments may be available. Examples here: yellow fever virus, plague, anthrax, Coccidioidomycosis. So those infections, they are treatable or preventable, or they don't spread easily. Does that make sense? But they can cause severe illness: Coccidioidomycosis, yellow fever, anthrax, plague, all of them. And finally, the coronavirus, by the way, is BSL 3 agent. Because it's very easily transmitted, there's no treatment or vaccine, although disease isn't that bad, okay, compared to, say, anthrax. BSL 4, exotic. I really cannot think of any original pathogen from the United States. Lethal illness. Now, when I say lethal, please take it with a grain of salt. Not 100% lethal, but very lethal, like the bull. Lethal illness, no vaccine, and does that make sense? So examples of BSL-4 pathogens are virtually all hemorrhagic fevers. They all are exotic. The ones that you've heard about probably Nipah, Ebola, Marburg, Lassa, Machupo, Hendra, all viral hemorrhagic fevers are BSL-4 pathogens. In the lecture notes, there's a link to a pretty cool video of what BSL-4 is like a tour, a film about BSL-4. It's really, really good. Does that make sense? We good so far? Okay. The next topic of conversation, levels of cleanliness, quote unquote. How can we categorize different sort of protocols for microbial control? So microbial control, we can target living tissue, or we can target inanimate surfaces. So for living tissue, asepsis, okay, usually through the application of chemicals. And degermination. It's mechanical removal, basically. For inanimate surfaces, we have sterilization, all microbes are gone. All microbes are gone, usually temperature or chemicals. We have disinfection. Some microbes are gone. Also by application of temperature or chemicals. And finally, we have third approach, sanitization. Okay, reduction of microbial number, usually mechanical, like dishwashing. Does that make sense? Are you following? Now, a couple of really important things here. So, grade of resistance. How are we gonna between different microorganisms? So, most resistant on top, least resistant on the bottom. So, most resistant, bacterial endospores and prions. Okay, one group. Kind of in the middle, protozoan cysts, sexual fungal spores. And naked viruses. And finally, least resistant protozoa, fungi, asexual fungal spores, and the localized. So I can ask you to compare organisms in terms of their resistance. Does that make sense? But I will not ask you to compare organisms that belong to the same group, so you won't have to compare, let's say, naked viruses to sexual fungus. Am I clear? Good? Okay. Now, another concept that I want to introduce. So okay. The concept that I want to introduce is called D-value. Okay, so imagine that this is sort of a survival graph of microorganisms. Okay. So this is number of cells. This is time. So this is how number of cells changes with the time. So your method, whatever it is, okay, kills microbes. Does that make sense? Are you following? So imagine that this number here is 100 cells. And this number here is 10 cells. So that's logarithmic scale. Okay. Now look at the time. You see that time? I'm going to call it D. D is called decimal reduction time, or D value. So it is time that is required to reduce the number of microorganisms 10 times. Does that make sense? From 100 to 10, or from 1,000 to 100, or from 10,000 to 1,000, 10 times. Decimal reduction time. Are we clear? Okay. Now, what it depends upon. Temperature, obviously, pH, pressure, presence of organics. Now, what do I mean by organics? For those, Grace, who work in the hospital, human excretions like blood, feces, saliva, mucus are protected. Does that make sense? So say survival of coronavirus by itself and survival of coronavirus in a mucus are two different things. So far, we good? Prions, Prions. Those are cysts, cysts. So far, good? Now, a couple more words about targets of different methods. Cell wall, cell membrane, proteins and DNA. Now look at this. Look, you disrupt cell wall, you get lysis of the cell. Does that make sense? You disrupt cell membrane, you get lysis of the cell. You inactivate, denature proteins and DNA, you get cell is dysfunction. Okay. Something else that I wanted to add about D value. Higher D value, more effective, less effective method, or higher resistance. Does that make sense? Look at this. The longer it takes to kill a microbe, the less effective method you have. The higher the D value, the longer it takes to kill microbe, the less effective method. Or the microbe has high resistance. Are we clear? Okay, so basically we can say that higher D value, lower. All right, so let's talk about some methods. So for methods, I promise I will tell you everything that you need to know. Seriously. You should be able to get an idea of when you would use a particular method, for what you would use a particular method, okay? And we're going to start with the physical methods, okay? And the first one will be temperature. So temperature, you can have methods that use, well, cold temperatures, methods that use heat, okay? Cold temperatures, obviously. Refrigeration. Now, refrigeration is not, it's a static method. Okay? Are you familiar with the expression bactericidal versus bacteristatic? So static means it doesn't kill microorganisms, it just stops them from growing. Are we clear? Freezing. Also a static method. Okay? Does not kill microbes. And lyophilization, right? So that's going to be also static. Now, all these three methods, as you can imagine, are crucial for food preservation. Does that make sense? Good? Okay. Now, heat mediated. So heat, you can have dry and moist heat. Good? Moist heat is always more effective than the dry heat. Okay? So D value for moist compared to D value for dry will be less. Does that make sense? It will take less time for the moist heat methods to destroy microorganisms. Are we clear? Okay, let's move on. Dry heat. Incineration. You know what incineration is? Burning. Simply burning. So it is a sterilizing method. You can't use it for everything though, right? You're not gonna sterilize this mug using incineration mainly because of the cat leak. Does that make sense? Dry oven. Sterilization with an asterisk. Some regimens are sterilizing, some are disinfecting. With incineration, you don't have such, okay? And desiccation. Okay. So remember we mentioned that the most resistant are endospores. So basically the crucial moment, is your method sporicidal or not? If it is sporicidal, if it can destroy endospores of bacteria, it's sterilizing. Am I clear? So desiccation is not sterilizing. We're going to put disinfection. But you gotta understand, we use it mainly for foods, like drying out foods. Okay, that makes sense. Good. Okay. Moist heat, autoclave, so pressurized steam, sterilizing. Boiling, disinfecting. Boiling is not sterilization. Pasteurization is yet another example of disinfecting method. Does that make sense? We're good? So for all these methods, we're talking about disrupting cell wall or denaturing proteins and nucleic acids. We're good? Any questions here? Okay, radiation control methods. Radiation can be ionizing and non-ionizing. Okay, so ionizing radiation, X-rays, gamma rays. These are sterilizing. Now the advantage of ionizing radiation is that it is very non-invasive. Does that make sense? The penetration capacity is highest in a gamma radiation. But you can sterilize food because you do not alter the chemical composition of food. Does that make sense? That's really important. Because if you want to sterilize the burger, can you stick it into an autoclave? Wait, you can. What comes out? Can you eat it? You can, just going to be very unappetizing. Does that make sense? So if you want to kind of keep food unaltered, that's the way to go. Does that make sense to you? Non-ionizing, UV. Okay, that's disinfection. Okay, now, this is a good example of what I meant when I told you. Be able to figure out what you're going to use for a certain purpose. Imagine I ask you, what would you use for disinfecting air in the dental office? You only need to disinfect, so UV light will do just fine. Does that make sense? You can't pasteurize air. You can't do dry oven for air. Does that make sense? If I ask you, what would you use for sterilizing surgical instruments? Sterilizing. So you basically have incineration, X-ray, autoclave. What's the most convenient? Autoclave. It's in every hospital. Does that make sense? Kind of follow this idea. Third method of microbial control, physical method, is filtration. Okay, so that's going to be for liquids and gases. Okay, so that's going to be membrane filtration. And that's going to be EBOT filtration, high efficiency particulate air. Now, can you sterilize? You absolutely can. It's only a matter of a price tag. So can be sterilized. Does that make sense so far? Good? Okay. So that's your physical methods of control. Now let's go over some chemical methods. And I'm going to group those methods loosely based on the underlying principle. So the first group will be oxidants. Oxidants. Halogens and peroxide. Okay, halogens. Fluorine, disinfection, sterilization with a question mark or asterisk, depending on the length and the dose. So if you take, say, concentrated bleach, pour it on something and leave it there for 30 minutes, yeah, it's sporicidal, but we don't do that regularly. Does that make sense? So chlorine, we use chlorine in the form of hypochlorite. There will be no chemical reactions. Don't worry about that. Okay. And iodine. That's asepsis. We use iodine specifically in the form of betadine for aseptic techniques, preparation of the skin. Does that make sense? Peroxide. Hydrogen peroxide. Disinfection. Asepsis. And sterilization if you use vapors. Does that make sense? Vaporized hydrogen peroxide is sterilizing, can sterilize rooms. Okay? And puracetic acid. It's liquid and it's sterilizing. Does that make sense to you? Protein denaturation and membrane disruption. Okay, so phenolics. Disinfection. Very toxic. So we use them not frequently in household items. Are you familiar with Lysol? It contains phenolics. Okay. Are you familiar with the name triclosan? Well, it's a new generation. Triclosan at some point was added to virtually every soap and shampoo in the United States because it was shown to have antibacterial properties. Later, it was demonstrated that triclosan not only has very kind of, weak antibacterial properties. Bacteria, since we used it so much, bacteria developed resistance to it. Does that make sense? Some people claimed that triclosan was toxic to humans. I mean, everything is toxic. In the concentrations that it was used for soaps and stuff, it wasn't. Toxicity wasn't an issue. An issue was that it was useless. It wasn't very active in the first place, and bacteria developed resistance. Am I clear? Okay. These big guanidins. Now, you see what I do here? Protein, that's the mechanism that they use. Protein denaturation and membrane disruption. Got it? These big guanidins. Disinfection, asepsis. Very safe and effective. If you are familiar with the term chlorhexidine, that's what I'm talking about. It's very safe, very low toxicity, very effective. Perhaps the best disinfection agent that we have for the hospital. Alcohols, I don't really know what to say. Well, they disinfect. Disinfection, asepsis. Okay, good so far? Surfactants, surface active compounds. Okay, surfactants can be cationic. It's called quaternary ammonia. Um, 90% of your household cleaning, quaternary ammonia compounds. Positively charged. Okay. Now, you remember the surface of bacterial cells is negatively charged, right? Membrane is negatively charged because of the phosphate groups. So positively charged quaternary ammonia are more efficient. They have a positive charge. I'm going to show it using my hands. A positive charge, quaternary ammonia, and a long organic fatty tail. So what happens, they stick to the membrane, they stick the tail into the membrane and disrupt the membrane structure. Does that make sense? So since they're positively charged and can bind to the negatively charged membrane, they're more efficient, more effective. Okay. Disinfection. Sanitization. So like regular cleaning. Anionic compounds. Soaps. Okay, negatively charged, meaning that they are less effective. Suitable for sanitization. Now I'm going to take a very short kind of tangent. Remember I told you I'm going to ask what would you use for blah, blah, blah. Imagine I ask you, what would you use for cleaning floors in the hospital? You have to take into account safety, price, level of cleanliness. Does that make sense? So if I ask you, what would you use to clean floor in the hospital? Don't suggest alcohols, please. Make sense? And I will not make you to choose between, say, a solution of quaternary ammonia, a solution of soap, and weak solution of chlorine bleach. All are suitable. Does that make sense? If I ask you, what do you need to sterilize the surface? Peracetic acid, for instance, right? Soap's not going to do it. Okay, surfactants, brilliant. Okay. Next one is alkylating agents. Okay, so we're going to group them slightly different. What they do, they chemically modify and denature DNA and proteins. Liquid alkylating agents. Glutaraldehyde, phthalate, metapropial lactate. Okay. Gases. Ethylene and propylene oxides. Formaldehyde. All of them are sterilizing. Okay, they all sterilizing. You can use them to sterilize gases. Look, gases, you can sterilize rooms or plastic that cannot withstand temperature. Does that make sense? Of course, they are very, very, very toxic for humans. No shit. Okay, does that make sense so far? Good? Grace, question? Okay, last bit for chemicals, heavy metals. Okay, so disinfecting. They usually disrupt various enzymatic pathways. Most commonly used is a copper and silver. Okay, zinc, its efficiency is with a big question mark. Gold, I mean, gold is very antimicrobial, but it's also very expensive. This is why we don't use it. Does that make sense? Now, they are oligodynamic. What it means is they can work in extremely small amounts, in minute amounts. Do you understand what I'm saying? Now, here's the deal. Listen up. There are some restrictions. You can't spray copper or silver. Does that make sense? So we use them to cover surfaces like doorknobs or faucets and stuff like that. Any questions here? Okay. Now, the very last bit. While I'm getting rid of it, again, when I will be asking you questions like, what would you use for sterilization of plastic? Okay, sterilizing, non-temperature resistant, okay? So we can't use autoclave, or obviously we can't use incineration. So we got to use either radiation or chemicals. Can we use gamma? Yes. Can we use ethylene oxide? Sure, no problem. Does that make sense? Last bit that I wanted to mention before, you see I have biofilms written there. So biofilms, I forgot to talk about them when we were talking about microbial communities. Biofilms, microorganisms grow mostly on the surfaces. These cultures that we have right there, so-called planktonic cultures, are rare. They grow on surfaces. So here's how growth goes. You have primary colonizers. These primary colonizers stick to the surface and start to produce extracellular polymeric substances. Basically carbohydrates and proteins. Essentially, this is an extension of glycocalyx of those bacterial cells. Okay, we're good? That makes sense? Extracellular polymeric substances produce, quote unquote, goo on the surface. This goo attracts secondary colonizers, and it also protects against desiccation and predation. Does that make sense? Now, secondary colonizers lead to increased density, okay, which reaches at some point what is called, its official term, critical density, which leads to production of so-called quorum-sensing molecules. Okay, so what are they? What they do? They regulate gene expression. And they actually can induce themselves. So this feature is called to be auto-inducers. Does that make sense? Now, let me kind of walk you through this in a smooth way. So imagine you have a surface. You have primary colonizers sticking to it. They start producing the gooey, carbohydrate-rich, extracellular polymeric substance, which forms a goo that protects the colony from desiccation and predation. Also, it protects from antibiotics and stuff. So biofilms are notoriously hard to get rid of. This goo facilitates the fusion of secondary colonizers. Good. Secondary colonizers lead to increased density in a biofilm. And when density reaches the critical density, microbes start to produce quorum-sensing molecules. Look at this. Quorum-sensing molecules, what they do, they start regulating gene expression in the colony. It may alter metabolic processes. It may start producing virulence factors, okay? Immune evasion. They also induce themselves. Does that make sense? I usually compare to how, like, imagine you have settlers in the United States. And if you are living, like if you are the only person in the state of Kansas, you really are not bound by any laws because you don't interact with many other people. Does that make sense? But then people start to live closer to each other and they have to start regulating the community. So this is what these quorum-sensing molecules are. And they're different between gram-positive and gram-negatives, I say about it in the lecture. Gram-positives produce oligopeptides and gram-negatives produce some certain lipid derivatives, which kind of makes sense because cell walls are different. So you got to have different molecules to get released from those cells. We good? Make sense? Okay, we're going to talk about antibiotics next week and look at the results of our amazing